we're going to be talking about inoculation and spore work and cloning. You'll gain the confidence in handling cultures and walk away with lots of information on agar, inoculation, incubation, and find out how to turn your liquid culture into 30 liquid cultures, find out how to clone and work with spores. And then for mission 3, we're going to learn about fruiting, incubation, and harvesting. So we are going to explore substrates, grains, incubation timing, fruiting chamber tricks, and how to troubleshoot issues. And by the end of everything, you should know how to harvest, how to store your own mushrooms, and have the confidence to keep growing again and again. So at the end of everything, you should have practical skills, some digital boosters, and you should get a certificate showing that you are officially ready to go. And yeah, I'm super
A badge too. Show them the
yeah.
badge.
Oh my gosh, yeah. Hold on. I don't know if you could see, but I've got the little sport course badge right here. I have it on my hat. We also got some stickers and we got some patches. It's going to be really, really fun. I'm super excited. So you guys should be too.
Yay.
Yay.
Super excited for it. And while this one's online, we're doing a kickoff party where we're learning how to build our own flow hood, which is like the key tool that you need for doing sterile mushroom cultivation. So join us if you can join us at this launch workshop on the 28th. And we have a bunch more events coming up. I was just doing a count the other day. I haven't gotten them all on the calendar, but we have 24 events coming up in October. I always tell people October, it's the month of Halloween. It's really symbolic for nutrient cycling, the Day of the Dead, Dia de los Muertos, like so many cool things happening in October. And it's a mania. It's a mania month. And so we have a lot of events on our calendar. Some of them we're looking for help, volunteers, but definitely keep an eye out for everything happening. We have a lot of block giveaways, a bunch of events happening with Roots and Wings. I think we're going to be at 4 different events. We're doing an event with the Central Library cooking club, like a cooking class. We're going to be doing some chicken of the woods tikka masala and other goodies. So if you want to learn about chicken of the woods and about culinary mushrooms, how amazing they are, how tips on cooking them, join us at the Central Library downtown Austin. We're doing another event with another branch of the library, and here in Austin we have Recycled Reads, where the library actually fundraises, and they have older books, but we've got some donated recycled reeds, and we're going to learn how to grow mushrooms on the books. So check us out there. Anything else? Yeah. So, and our guest speaker is coming all the way down next month, is going to be coming from Boston. She's a writer, a poet. She's coming out with this book about how she learned to grow and become how fungi taught her. So it's a little bit of a biography, but I think it'll be a really great event. So we have a mix of online and in-person events. But tonight, our focus is going to be on what if fungi win? And I put a hash bang because I was already like, yes, they do win. At least that's how they already won. I have to tell people all the time, like, will mushrooms save us? And I was like, they have saved our planet already, like through every mass extinction. So they have won many times. And that's why I like the hash bang behind this, behind the word win. But I'm super excited for you all to meet and learn from Dr. Arturo. I learned about him years ago, and I have a fascination with, I was telling him earlier, like I, was an artist, designer, animator prior to becoming a citizen mycologist. And I was fascinated with like pigments and color in plants and fungi. And this last summer, or I think it was even the spring, it was during the City Nature Challenge in April, we found an albino wood ear. And I was like, I know there's a mycologist out there. And I had read some of his papers and listened to some of his talks. And there was some more current research and things that he was sharing that I found online. So I reached out to Dr. Casadevall, and he's so gracious to join us tonight and share some of these fascinating facts about fungi. But if you don't know if you haven't read his bio, comes to us from Johns Hopkins and is one of the foremost experts on fungal pathogenesis. He's authored over 1,000 scientific papers, led groundbreaking work during COVID-19 pandemic, and has written extensively on the future of science and public health. And tonight we're also going to be doing a random giveaway. We'll do that at the end at the Q&A. We're going to be giving away a copy of this awesome book that he has out. So everyone, please join us in a warm welcome for Dr. Casadevall.
Thank you. Thank you so much, Angel. I think, as I was telling Angel, I think the discovery of that albino wood ear is very interesting because the fungi may be losing their pigments in order not to overheat as the climate gets warmer. And maybe you can all keep your eye out for it, because documenting something like this is not going to be done by the establishment. It's probably going to be done by citizen science, and potentially you can get that information when you're looking. So I thought that we would have some fun. So what I picked out of this book are some highlights talking about the world's largest organism, our closest relatives, a cold kingdom, melanins. We're going to talk about an idea that fungi are responsible for us. And finally, the fungi and global warming. So I'm sure many of you know that the largest organism in the world is a fungus. It's in Oregon. It's about 3.5 square miles. It's growing. And the reason you know it's one organism is because people have been collecting the mushroom, the scientists bring them back to the lab, sequence them, and it's the same organism. It weighs as much as a World War II battleship, and you can tell where it is at because the trees above it tend to be quite sick and dying. They are our closest relatives, so they are a lot closer to you than plants or protozoa, for example, and that's a fascinating story that is not well understood, but somewhere about 1 billion years ago, the fungi and the animals split. And the animals split much earlier. Now, this is a problem for medicine because it means that we are so closely related that it's hard to find antifungal drugs. Therefore, we don't have that many. It's a cold kingdom. So during COVID, Radamés Cordero was my lab, took a thermal camera, and walked in the woods here in Maryland. And he noticed that when he pointed the camera to any mushrooms, they were blue, while the rest of the environment was either yellow or red. And that's because they were colder than the environment. And so we immediately began looking in the literature, and people had documented, and I'm sure you have that experience, that mushrooms are cooler. So we had the idea that maybe after looking at many species, that maybe we should look at the microscopic fungi, the ones that grow in agar plates. And he looked at multiple species. And the amazing thing was that even the microscopic ones work really hard to stay cool. So if you look at the right, you see that there is a square and there is a temperature gradient on the right across a colony. And what you can see is the agar colony is about 30 degrees, and then it gets a little warmer, and that's because it's releasing enzymes that's breaking up the nutrients, and then the colony drops. Even when you put these colonies in a refrigerator at 4 degrees, they stay colder than the environment. The fungi want to be colder than the environment. And what is the mechanism? The mechanism is the same as why are you cold when you walk out of a shower when the bathroom is steaming? The reason is you have a little layer of water that is evaporating, and that makes you feel cold. So as long as they have water, they stay cool. And then we had a lot of fun. We made an air conditioner, a prototype, in which went out to the store on board mushrooms, and it can cool for about 10 degrees centigrade, as long as the mushrooms have water, and then afterwards you could eat it. We submitted this as a patent, and it's the only patent in my life that came back with accepted right away. And I think the examiner took a look at this and said, my God, this is so different. I don't really have to worry too much. Fungal leather is coming. Fungi can be made to grow in multiple shapes, into a hat, into a purse. And this material, it feels like leather. And in fact, there are companies that are making it. It's primarily been seen in Europe, but it offers a completely different option than the skins of animals. And I think that fungal leather has a future. Now I'm going to talk about melanin, which Angel briefly mentioned. The word melanin comes from the Greek, or melanin being black. It is a pigment that is usually dark, can be black to brown. So when you see mushrooms on the ground, they're brown, they're even if you turn them over and they're black, that's melanin. And it's the same melanin that is in your skin. This is melanins are not proteins, they are pigments. So they are made by most, if not all, fungal species, and they've been associated with numerous functions. But the most interesting thing is that they are resistant to strong acids. So if you were to take your favorite mushroom and boil it in hydrochloric acid, everything will disappear except for melanin.
Sorry,
Okay,
I accidentally muted you. Sorry about that.
it's all right. Allow me to unmute myself, which was kind of nice. So the bottom line is melanin is really, really resistant and tough. And let's see what this is in. So it's, melanin is everywhere. It's in your skin. It's in the ink of the cuttlefish is melanins. If you look at the banana there on the right, that is trying to repair itself with a melanin patch. These are remarkable pigments that are found in all walks of life. And here is a fungus I work with. If you give it L-Dopa, it turns completely black. Where is the melanin? The melanin is put in the cell wall of the fungus. So you're seeing this a microscopic fungus, and it's got a really complex way on which the fungi do this in cell biology. It is made inside the cell and is shipped to the outside. That is important because by being on the outside, it's in a position to interact with the environment and to protect the fungus. So this is what I was mentioning before. If you take these organisms and you grow them in L-Dopa, L-Dopa is like food for melanin. They make a lot of melanin. You can recover these ghosts, these shapes, which are the same shape as a cell. But if you are not melanized and you put them in concentrated hydrochloric acid and boil it, it all goes in solution. So melanin it's when you look at it, it's composed of these small granules, and it serves as some sort of armor. It's not it's not full made, it's not chainmail, but it's more like somewhere between studded leather and chainmail, because it's got to be able to allow the nutrients to get into the cell. It protects against practically anything you can throw at it. Heat, enzymes, oxidants, defensins, which are small molecules, proteins that kill fungi, heavy metals, and as well as antifungal agents. So one of the problems physicians have is that these diseases are very hard to eradicate. And some of the issue is that they make melanin in tissue, and this gives them acquired drug resistance. So the story of melanin is far more interesting than just looking at what it is telling. This is a picture of the Chernobyl reactor. It was a nuclear explosion. It was a chemical explosion in 1986. And what the old Soviet Union did was to build sarcophagus around it because they needed to keep the other reactors going. In fact, Chernobyl has been in the news because Ukraine is still running the other reactors. So it looked like this thing was going to collapse around the year 2000. And the European Union sent a robot in there. And that picture you see is the robot. Look at the walls. They're black. And they're not black because of smoke or fire. They're black because they cover with a pitch black fungus. And we were very interested as to what was going on there. Could it be that this melanin was protecting against radiation? And to put you in perspective, if you walk in there, you are lethally irradiated after 5 minutes. The only way to survive would be a bone marrow transplant. Yet this fungi have no trouble growing in a high radiation environment. So we looked at it, and the fungus does protect against radiation. It's got about half the shielding of lead, that is the melanin. That's pretty significant. But we decided that this couldn't be the only explanation because fungi are naturally very resistant to radiation, even when they don't have melanin. To tell you a little bit more of that story, if you take black mushrooms and white mushrooms right there, and you feed them to a mouse, and then you lethally irradiate the mouse, you can protect the mouse against radiation, even though the radiation is destroying its bone marrow. How can that be? Well, the mushroom, the black material, coats the gut and protects it. And if you protect the gut, it doesn't leak bacteria. And also there are lymphatic organs that then seed the rest of the mouse. So you see that experiment at the top. The black mushrooms or the white mushrooms for synthetic melanin survive, whereas the ones that got just saline or white mushrooms alone all died. There's a lot of interest in this, obviously, for treating radiation. When we send the information to Japan when their reactor went, but I just want to point out that this is being reproduced by other groups. And if you are near a nuclear disaster, probably a good idea to eat as many black mushrooms as you can get your hands on. There's more to this story, and that is that these fungi from Chernobyl grow towards radiation sources. It's an amazing thing that it's been called radiotropism. So if you put a radiation source in the agar, the fungi grow toward it. And in some ways, they wrap their hypha around it. So we began to think that maybe they were using the melanin to collect radiation for food. And we spent several years in this project and in fact showed that the melanin will drive the oxidation reaction of NAD, which is an important energy molecule, to NADH. This was done with my collaborator at the time, Katerina Darachova, and showed that this could lead to biological useful energy. Let me tell you a little bit about science. We found this really interesting. So we sent it to one of those fancy one-word journals. And they took it for a year and they gave us a lot more to do, and then they rejected it. They just didn't want to handle this. So we went to the next journal, and again, the same situation happened, but we had 3 reviews that I wish I'd saved because they capture science so well. The first person said, look, this is really interesting. Why don't you publish it? Even if they're wrong, this is going to stimulate a lot of science. Reviewer number 2, though, wouldn't let it go unless we could track an electron all the way from melanin to ATP, which is and that you can't even do that for photosynthesis. But it was reviewer number 3, the one that we couldn't argue with. Reviewer number 3 says, I can find nothing wrong with the work, but I don't believe it. So what do? Then you go to the third journal, and we managed after a while to get it published, and today it's one of our most cited papers. The interesting thing is that the journals that did not want to publish it had to write articles around it because there was an enormous interest when it happened. And it just shows you the discontinuity between what is science news and what can get published. There was one criticism of the work that bothered me. Somebody said to me, Arturo, X-rays go through tissue. Gamma rays are far more energetic than X-rays. How could the melanin be capturing gamma rays? And I said, oh my God, we must have made a mistake somewhere. And we spent 4 years just to demonstrate that fungal melanins can interact with gamma rays. Gamma rays are enormously energetic. And we did it by various ways, but the actual proof came from this man called Charles Turek. He made a melanin electrode, and he put it under a gamma ray beam, and he got a current. And by that, it's unequivocal that the gamma rays are interacting with melanin. You don't get a current if there is no melanin in the electrode, and that this is happening probably by some sort of resonance that we don't really understand. It's also been confirmed by other groups, independent groups at the United States Naval Research Laboratory. And here may be some real world consequences of that. The space station is overrun by fungi. And it's a major problem now because this stuff grows wild in space. Space is a high radiation environment. And you can see there a door that's got some sort of black mushroom growing on it. You see electronics where the mushrooms grow in it. And there is really no way around this because every time you send up a relief ship or to bring supplies, once they open the door, you bring in fungal spores. So I think that when it comes to astrobiology and humanities, future plans in space, we're going to have to deal with the fungi because they do very, very well up in zero gravity and high radiation. In fact, NASA is very interested in this, and you can go to the NASA site and find that they have something called mycotexture. They're trying to think, how can you use melanin? Fungi and melanin for space colonization. The beauty is that you only have to send one spore, right? You don't have to send the materials up. And if you do it just right, the stuff can grow in place and can give you your mycotexture. This phenomenon is probably responsible for the Black Mountains in Antarctica. The Black Mountains of Antarctica are black because they are a symbiosis between a fungus and an alga. And what you can see there is that these are probably the pigments are collecting radiation and generating food for whatever is growing on the side. Now, one consequence of this phenomenon, which is very bad for us, is that whenever glaciers begin to melt, holes appear that are filled with melanotic organisms that then collect more energy and rapidly drive the cycle of glacier melting. So this phenomenon, it's got a lot of consequences from astrobiology to what is happening here on Earth. I want to introduce you to a new concept that we developed, which is called disaster microbiology. So whenever you hear about a disaster, you think about what happens to what you can see. The structures are damaged and all that. But what people don't realize is that the entire microbiome of the area is altered, and that these changes to fungi, bacteria that happen with the disaster complicate the situation tremendously. Think about Katrina. What destroyed New Orleans was not so much the water. When the water came down, they had to tear down all those houses because of mold. Had there been no mold, a lot of that could have been repaired. And I want to talk a bit about global warming. I mean, I think global warming is a fact. I mean, people are recognizing that this is the way. There may not be agreement as to what's causing it. But I think we're living at a time of climate change, and we are the first humans that have ever experienced climate change. Because in the past, it happened so slowly that no one person could have seen the climate change. And when this happens, it's going to affect all aspects of life, the life that you can see or the microscopic life. And give you an example of that, I take you to the Permian. The Permian was an epoch that ended about 255 million years ago, and an event happened that wiped out about 90% of life. The current understanding is that there was a coal fire in what is now Siberia that burned for 80,000 years that was associated with climate change. Basically, as the coal burned, it increased CO2 and reduced oxygen. And this was published in PNAS a few years ago. But the idea is that it slowly changed the climate and led to the acidification of the oceans and to just the destruction of what was the Permian flora that had been around for millions of years. But guess who does well? As Angel said in the introduction, whenever the earth has a bad day. This is a mountain in South Africa in which there is a 1 meter of fungal spores separating the late Permian from the early Triassic. I mean, think about that. Enough spores deposited 255 million years ago that you can still find the evidence of that enormous fungal bloom. What that means is anything that survived had to deal with the fungi. And right now, this is an area that it's increasingly recognized, but it hasn't really been taken so much into account. And later, I'm going to tell you about another fungal spike that I think is responsible for us. The Permian fossil resemble modern plant pathogenic fungi when you look in the microscope, suggesting that any life that survived, whether plant life or animal life, would have had to deal with a massive proliferation of these organisms. The fungal kingdom, I'm sure you all know it better than I do, is the largest kingdom, and it includes the major pathogens of plants, insects, and vertebrates. They're currently, or cold-blooded vertebrates. They're currently devastating major ecosystems, the white nose syndrome of bats that I'll mention in a minute, the catastrophic amphibian declines, the salamanders in Europe, the snakes in North America. But mammals are remarkably resistant. People worry about bacterial infection, viral infections, parasites. They take prophylaxis when they travel to a place with malaria. But you don't hear people worrying that they're going to die of a fungal disease, even though they worry about fungi in the nails and athlete's foot. We don't really think of them as something that will kill you unless you get immunosuppressed. And on top of that, there are relatively few fungal species that are pathogenic for humans. These are the major ones. You can probably say, if you know 10 of them, you know 95% of medical mycology. So I ask you the question, why mammals? What makes us so special? The origin of mammals goes to the Permian. So they made it through that fungal proliferation. But then they stumbled in what is known the Triassic and the Cretaceous. It became the age of the dinosaurs. The mammals are different in that they have maintained high temperatures. So they have a stable internal environment. They can move and digest food during cold weather. If you see what happens in Florida whenever they have a cold spell, the iguanas can't move. And I will argue provides great protection against infectious diseases. Here is an example of the energy requirements. Here on the left is a log plot. So that's 10 by 10 changes. The elephant, us, the mammals need about 10,000 times the amount of energy on a weight basis than a fly needs, 1,000 times more than a frog needs. And that's because we spend so much energy maintaining our temperatures. So the great mammalian radiation begins at the end of the Cretaceous. And that's given us everything from whales to monkeys to bats. We live in the age of mammals. When you drive around Texas, what you see are birds and mammals. You don't see any large reptiles. They've all been essentially replaced. And what is the evidence that endothermy protects against fungal diseases? Well, this is an example of the temperature to inhibit urban fungus. He's doing a study here in Baltimore. And what he showed was that if you collect the fungus from the sidewalk, they grow pretty well at 30 degrees, but raise them to 42 degrees. And the only one that grew was Candida tropicalis, which is known to be a pathogen and to be temperature adapted. Today, the disaster is happening to organisms that are cold-blooded, whether they are the snakes, the turtles. The fungi can go right through the turtle egg and kill baby turtles. But frogs can be safe if you make saunas for them. This was a paper that appeared last year, and what some people have done is take cinder blocks and just attach them to a heat source. The frogs all jumped into them and spend their time there, raise their temperatures, and then kill off the fungus. And human fungal diseases are, in fact, if you're not immunosuppressed, the fungal diseases that people get are nail fungus, athlete's foot, dandruff, which is a fungal infection, and babies get fungal diaper rash. These are the cooler areas of the body. Some work has been done experimentally. You take a bunny, and you take a fungus that is high pathogenic, like Cryptococcus neoformans, and you cannot give a fungal infection. The bunny is too hot. You can, however, give the bunny a skin infection or a testicular infection. But if you give the bunny steroids that depress the immune system, temperature is not enough, and you get a dead bunny. So the great resistance that we enjoyed is the result of higher temperatures and advanced immunity. So today, medicine often, medical treatments like transplants lower immunity, and that's why you get more fungal diseases, because heat is not enough to keep them out. In the United States, the white nose syndrome began in 2006 and is decimated North American bats. The bats are fine in the summer, but in the winter, they hibernate because there are no insects. They drop their temperature to 10 to 12 degrees, and then a cold-loving fungus grows on them. And they wake up in the middle of the winter and they die. If you take the infected bats back to the laboratory and just feed them, that will raise their temperature and they can clear the fungal infection just by going to 37 degrees. So some time ago, we looked in these, where are most fungal species? What's the maximum temperature they can handle? And most fungal species can go to about 30 degrees, which is ambient centigrade, ambient temperature. But between 30 and 37 degrees, that's our temperature, they decline rapidly. And this data came from a collection in Europe. We looked at 4,000 fungal species. And my friend Aviv Bergman is a mathematician, and there is a well-known formula to calculate how much energy do you need to be warm. So if you are 37 degrees, which is equivalent to 98 degrees, how much energy do you need? If you make your person colder, if you make the patient warmer. That's why fever, for example, often results in weight loss, chronic fevers, because you need so much energy to heat yourself. So he solved these two simultaneous equations. And the amazing thing was that our temperature, which is 37 degrees or 98 degrees, seems to be perfect for keeping out the most fungi and not having to eat all the time. And to me, that suggested that something in our past selected for this temperature. So going back in time, there was the Cretaceous. That was the last time there were dinosaurs on Earth. And then something happened that we're going to talk about in a minute, but there was again massive fungal proliferation at what is known as the boundary between the Cretaceous and the Tertiary. And notice what happened. The dinosaurs disappeared, and then becomes the age of mammals and birds. The crocodiles seem to have made it through this cataclysm. So what the question many people say is, what killed the dinosaurs? Well, it depends who you ask. For a long time, it was thought that maybe it was small mammals that ate the eggs. That makes no sense. This makes no sense either. They did not go extinct from smoking. They could have gone extinct by increased volcanism. That there is some evidence now that the world before their end really was having a lot of volcanism, and the climate was changing very rapidly. But most authorities think that what really knocked them out was a rock from outer space that hit close to Texas. It hit Yucatan. There are a couple of explanations in the literature. I'll share you one. This is a very respectable journal. I don't believe this for a minute, but some people calculated that they could have passed enough gas to affect climate, and this is published. And the next slide, I'll present it to you in silence, because as a scientist, I have no data for or against. Oops. So the Cretaceous world and its end, it was a warm volcanic forested planet. The Atlantic was tiny, and this thing hit the Yucatan. And it essentially wiped out the reptilian megafauna. So there is fossil evidence that this thing led to global deforestation. Most of North America saw mile-high tsunamis that raced through North America, knocked down the woods, knocked down everything. And then the fires began because a lot of the Earth gets dumped into low orbit and then re-enters as flaming meteorites. So there is evidence for deforestation, massive fires. There is the global temperatures dropping one day by 10 degrees, and photosynthesis is shut down for 6 months, at least. And there is also evidence that the fungi, just like in the Permian, proliferated tremendously. So you had a lot of dead vegetation, and with dead vegetation, you got fungal proliferation, and fungal proliferation would have resulted in massive numbers of spores. And guess who would have been less susceptible? The small mammals, with their higher temperature, would have been resistant to fungal disease. This is just a schematic to show you what happened. The world prior to the asteroid you had conifers and flowering plants. And then what you see at the bottom is iridium that was brought in by the asteroid. And then you have this fungal layer where you see nothing but fungi. And eventually the plants recovered. So survivors would have again to deal with massive fungal proliferation. The post-impact world, what killed the dinosaurs was probably the blast. I don't think they got killed by fungal diseases. But the reptiles that survived fight off fungal infections with induced fevers, but there was no sun. Reptilian eggs are susceptible to fungal diseases. And in fact, today you can find dinosaur eggs that are invaded with fungi. So the idea was that a fungal filter at the end of the Cretaceous selects for you and me, the mammals, an endothermy. And this is published, and it's been sitting out there for a while. And it's a relatively simple idea. You had some survivors. We have iguanas today. We have salamanders today. So the survivors will have been small mammals and small reptiles. And then this fungal bloom happened. And between colder temperatures where the reptiles couldn't move as much, well, the small mammals had their internal combustion engines. They could go out there and forage for whatever food was available. That resulted in much larger populations. And when the skies cleared, then you had these populations went on to give you the great age of mammals. They essentially differentiated into the 4,000 mammalian species that we know. So this is published, and it's been a lot of fun to write, and I update it every few years as I learn more and more about geology and what happened. This idea has a lot of explanatory power. I haven't had any pushback. Now, there are two possibilities. People don't care about it. The other possibility is that, people just, well, that's Casadevall. Let him think whatever he wants, But it does provide a selection mechanism for a costly lifestyle. It explains why most mammalian temperatures are in the high 30s. It explains the tremendous resistance of mammals to fungal disease. It argues that your temperature is a major host defense mechanism. And it explains today why whenever you're sick, the doctors or anybody who cares for you gives you Tylenol or Motrin, because most of the things that we get infected with are already temperature adapted, like COVID. It could be a bacterial infection. You get them from other people. But if you go out into Texas, the wilds, and you fall on the ground and you get a wound and it's contaminated, a couple of degrees of fever could be the difference between survival and non-survival. Now, the fungi are adapting to the higher temperatures. We know that already. In fact, and there is an experiment here on the right done by a company that was trying to get a fungus to be a better control for insects. Whenever the insects are infected with this fungus, they find out a warm rock and they sun themselves and they kill off the fungus. They were able to train the fungus to grow at 37 degrees after a few weeks by simply incubating at a higher temperature. What he's telling you is the fungi have tremendous capacity to adapt. And we found evidence, although it's noisy, the bottom two lines are Basidiomyces. This is looking at the maximal temperature in a collection in Europe. So whenever you send something to a collection, somebody grows it at multiple temperatures, they recorded that. So we look at what was the maximum temperature for the Basidiomyces. And it looked like an upward trend beginning in the late 1990s. You don't see that with the Ascomyces. The Ascomyces are already temperature adapted. So we looked at Baltimore. Cities are heat islands. The temperature in the cities is about 5 to 8 degrees centigrade warmer than the environment. And we asked the question, could you document this type of rapid evolution in a city? Well, there was already a paper out there suggesting that this may happen. These are fungal species in which somebody found them in the city and found them in the areas around the city. And they showed that the ones from the city were much more likely to deal with heat shock. So that paper sat out there and we said, can we reproduce this? So we went to Baltimore, which is a city that gets really hot in the summer, probably not as hot as you do in central Texas, but the number of days that are hotter is increasing. And this is a heat map of Baltimore. And what the green areas are cooler, the red areas are hotter. And there's a streak there of the hottest city, the hottest street in Baltimore. It's known as Fayette Street. And Daniel Smith in our lab began to collect the fungi that he found on the sidewalk and in the area and bring them back into the lab and say, what is going on? So he asked the question, are urban fungi more temperature resistant? Are fungi in warmer neighborhoods more thermotolerant than those in cooler neighborhoods? Are fungi, the species, different? Well, the first thing he did is he noted that the fungi from the hotter neighborhood are more likely to be white than the ones from the cooler neighborhood. So they're losing their pigment. The stuff is so hot that you're selecting for more lighter color fungi. They may not be albinos yet, but they're getting there. So this is happening within 4 miles. The same species are changing. And then he looked at a Roloturul mucilaginosa, which is a known opportunistic fungus. And what he noticed was that the organisms from the warmer neighborhood were a lot more temperature resistant, that is to kill them, than the ones from the colder neighborhood. So this species has adapted within one city at a location that is 4 miles from each other. So some years ago, I argued that global warming was going to bring new diseases. Why? Because if we are protected from the fungi by a temperature gradient, what happens if the fungi learn to grow at your body temperature? And then there was this thing that came out a few years ago of Candida auris. Candida auris is a new disease to medicine. and appear, and I'm not going to tell you, appear all of a sudden. And some fact, it was first isolated from the ear canal of a patient in 2007, and it's Oris. People then said, oh, we must have missed it. So they went into all the collections, and they could only find a couple of isolates that were seen to have been misclassified. And then in 2009, 10, 11, and 12, you have simultaneous emergencies in 3 continents. That is, patients in hospitals began to come down with this. And the isolates were different from the different continents. Since then, we have had 3 more eruptions, one in Russia, one in Iran, and most recently last year in Singapore. And the isolates are not related. So what you're seeing is this is coming out of somewhere and is becoming a medical problem at that site. So the question is, how did it emerge independently in 3 continents? Well, there are 3 theories out there. It was there all the time, but people missed it. That's not the answer, because people have done retrospective analysis. It was a consequence of the overuse of azoles. Azoles are used in medicine and agriculture. So these are to protect the crops. Companies and farmers put in antifungals. The problem is it's unclear how antibiotic selection will give you a new virulent species. And then we propose that this was the result of climate change, that the fungus had adapted, and by adapting, that they have defeated the thermal barrier. And the problem is that is a speculation, conjecture, and circumstantial evidence. So we try to get evidence by looking at near relatives. What happens if you go into the environment and you find the ones that are nearly relative? It turns out that they are not as temperature adapted as Candida auris, consistent with the fact that something happened to this that changed it sometime in the recent past. And then my friend Anuradha Chaudhary in India managed to recover in the environment. She recovered it first in the South Andaman Islands. And what she showed there was they found it in 2 places, a beach where people used to frequent and probably contaminated the beach, and a marsh. And the isolates on the marsh could not grow very well at 37 degrees. So that is consistent with the fact that the wild Candida auris that hasn't adapted by passage to humans still adapting. And this could account for the continued emergencies. Now, to add to the problem, we are getting colder. You are colder than your great-grandparents. How can that be? Well, it turns out that a remarkable study was done comparing temperatures over 100 years. Now, temperature is something that we can measure, and we may be able to measure since the 18th century. So this is not a problem with thermometers. What happens is that we live in an increasingly clean world, and our grandparents and great-grandparents had a lot more inflammation. And they live in lives of greater inflammation. And that inflammation, they had worms, some carried TB, and that basically raised the temperature. So now we have a problem. The fungi are adapting to higher temperatures, and humans are getting colder. And I think that is a collision in the making. To add to insult to injury, once you expose fungi to higher temperatures, you greatly drive up mutagenesis. Most mutations are deleterious, but occasional ones will raise virulence and make them drug resistant. So they are not only adapting, but this adaptation is coming with increased genetic diversity. So I think the next one is my last slide, and I can't end without talking about The Last of Us, because I think The Last of Us did more for mycology than a lot of 100 years of research. Everybody's heard about the fungi now. And people, when this first came out, I found out about it. I didn't see the series because I've been getting emails. People say, Arturo, did you advise them on the crop? I had no idea what they were talking about. It turns out that I didn't, but a lot of reporters tracked me down. And the question was always, Dr. Casadevall, could this happen? And my answer was always, it's improbable, but not impossible. It's improbable because I don't know any fungus that can turn people into zombies. But it's not impossible because we know that a fungus can turn into an animal into a zombie, which is an ant. And it's not impossible because fungi can make compounds to make you hallucinate and can make you take a trip. LSD is made by fungi. So is there a fungus out there that we don't know about that it's got a compound that can be made in you that'll turn you into a zombie? I don't think so, but I can't rule it out. And with that, I want to thank you for listening, and I'm happy to chat about anything you want to chat about.
Yes.
So
Wow,
next
what a
question.
great talk. That was a deep time travel. We went to space.
Went to Spain, went to Chernobyl, went to the past, went to
What a
fungal
trip.
leather, took you to
What a
fungal.
mushroom trip that was. Great talk. One of the things I just wanted to kind of have a discussion point really quick, and it got me thinking, and I know Dr. Balan from the University of Houston is on the call too. So I don't know, Dr. Arturo, are you familiar with our state mushroom? It's
No,
the Ascomycetes. It's very
no.
exciting. How many people here know about our state mushroom, the Texas star, or have been on one of our walks?
Some
So
hands
it's
went up.
Coreactrus giester. It's this. It's the only mushroom in the genus. It's found along this kind of geological formation called the Devil's Backbone. It's also called, the mushroom's also called the Devil's Cigar because when it opens, like a lot of cup fungus, it makes an audible hiss and
Wow.
makes this hissing noise, like, and puts out this smoke or spores, right? And I hadn't like thought about it for, I talked to like a few mycologists and especially like soil people, like, why is this mushroom only in Texas? And then it was randomly found in a few other places in Japan and Taiwan.
Mm.
And they're very geographically distributed. And it's not like, oh, they were locked at some point and are, and during the tectonic shift, right? This biologist, when we did a walk at the Inks Lake State Park, I learned this was the oldest rock in Texas. It's where we got our pink granite to make the Pink Dome, which is our state capital. There's a beautiful, enchanted rock here that everyone likes to go rock climb and go see. It's a beautiful, beautiful part of our geology here. And talking to like some soil people, and I haven't had a chance to really like dig into this with geologists, but hearing you talk about these like mass extinction events where there's like all of these spores, it makes me think, just knowing where this sits in the larger phylogenetic tree of life, I feel like this mushroom was like maybe here a long time ago, and it came back with an uplift. And I would love to like do some collaborations with some natural science, geologists, people in that field, those fields. And we're wanting to like raise some funds and do like the full genome sequence, because Harvard did a study back in 2004. The technology was not, they were kind of comparing the specimen from Japan to the one in Texas. But it would be just amazing to kind of think about it from this kind of deep time angle.
The price of sequencing, as you know, has gone really down. I mean, we sequence Cryptococcus for $100.
Mm-hmm.
I mean, when you think that it costs millions and millions, the original genome project.
Yeah. But yeah, this
I greatly encourage you.
Highly melanated too.
I think that a lot of these questions can only be approached by citizen science. There are just not enough scientists, and they're wrapped up with other things. But you pose a question. I talked to the mycology group in Washington, D.C., and they were very interested in the idea that they go out on the weekends and they take pictures and they share with each other. I said, start a repository. start mapping where these things are. We may learn a lot as to what the biodistribution is if you do this a few years in a row.
Mm-hmm. Yeah. Well, how about we do the book giveaway first, and then we'll take some more questions. Let's see. So I picked Sherry's iPad was the random number. And I don't know if Sherry's still on, but I think I might actually know who this Sherry is too. I'll look at the registration list and email Sherry's iPad.
Sure, it's like,
Yeah, so we'll get the book out. Woohoo, Sherry's iPad. So other questions. Steve, I know you've got a question. You're a doctor.
Fascinating talk. I don't have a specific question, but
Yeah, just
it
a
is
lot of
about radiation stuff.
wonder.
I need to start eating a lot of black mushrooms.
Yeah, Dr. Balan, do you want to unmute?
Yes, Dr. Arturo, it was a fantastic talk. I have learned a lot of stuff from your presentation. I was fascinated with your discovery that fungus can harvest the energy, high energy radiations, and grow on it. And, do you think, because the world is going towards a lot of malnutrition, hunger, poverty, do you think how this discovery can turn into abundant food resources for to eradicate poverty and hunger? Have you thought about it?
so Dr. Balan, we published this already like 15 years ago, and it was confirmed by another group, and it's never really been picked up. And I think part of the problem is that science has a lot of trouble with things that are interdisciplinary. The high energy physics don't work on biology. The biologists don't think about high energy physics. And that's been kind of the problem. But the people who are really interested and that kept some of this going is NASA, because clearly the implications are enormous. It means, by the way, this fungi will have no problem growing in the soils of Mars.
Okay.
So if you try to think about a future in which we're going to colonize some of these worlds, the fungi can help us, prepare them in some ways, losing minerals and something like that. So one day you could have crops.
Really fascinating work. Really appreciate your presentation.
Thank you.
Yeah. Is any of the papers that you've published look at melanin and kind of like cleaning up toxins in soil?
I haven't done it, but other people have done it. This stuff is like activated charcoal.
Okay.
So in fact, people have designed some clever, what I call fungal melanotic filters, in which you grow a mat of black mushrooms and you pass the water through it, and the melanin basically acts like the filter in your aquarium. It picks up
Yeah.
a lot of these lifelong chemicals and things like that. It pretty much absorbs anything because so yeah, there is interest, but again, you find it in the internet. You go in there and you put down fungal filtration water system, and you find that people have done things like that, but it hasn't gotten to the point in which you can go to the hardware store.
Yeah. Or is it something that
there is a cartridge or something that you could put in, so
yeah,
to your
or
water.
used like in an industrial scale, like water treatment
Exactly.
plants and things like that? So Tommy asks, is black mold that grows in space stations considered an extremophile?
I guess so. I don't know. It depends. It depends how you define strong, because the people are in there too, right? It's not like we are in an extreme environment. The environment is good enough for the astronauts, but this is kind of growing in that. I consider the fungi extremophiles. I mean, if you can, certainly the fungus is growing in Chernobyl. I mean, that is a very inhospitable environment.
Okay. So Veronica asks, plants are susceptible to fungal infections. What do you think are the implications to our big use of fungicides in agriculture?
Well, people ask me that because with the problem in the Netherlands has been that they were using the fungicides to protect the tulips.
Mm,
And what was killing the tulips doesn't kill people, so they weren't worried about it until aspergillus began to turn up at the local hospital that was resistant to the
mm,
drugs that are very similar to the fungicide. So in other words, you spray the field, you take care of the plants, but you're also spraying everything that is in the soil. And that could end up selecting for it. So I think it's a hard decision because we got to eat.
yeah.
I mean, what is the choice going to be between, treating a fungal disease and eating? I'll take eating.
Hmm. Yeah. So Keith asks, would the loss of melanin with higher temperature mean that black fungi be less able to deal with radiation?
It would be. It would be. I mean, but yeah, I mean, I think that is a trade-off. I don't think they eventually lose the capacity to make melanin. They just select for ones that are making the less. I think that if you again challenge them with radiation or something, you may get the melanin back.
Hmm. So Jamie asks, what is the lower temperature range of fungal growth?
I don't know, but there is fungus in the thermofrost.
Hmm.
So I think that you just slow it, but I mean, look, they grow pretty well in your refrigerator, right? When you leave something in there and it's just an example. I think they are remarkable. I don't know what the lower level is. I don't know if somebody's basically put them at -20, -40 and see, but they're not killed by the cold. I can tell you that.
Hmm. Yeah, they like that cold shock for sure. Are there any other questions? I know I was thinking of several, especially when you were getting into the Jurassic Triassic extinction event. Um, there's just so much back and forth debate about kind of what caused it, but also the evolution of fungi.
Absolutely.
Um, like how all of a sudden and was it, we don't know, like, was it, did fungi evolve because all of a sudden there was just all this food and it was like, let's eat all of the parts of wood, the cellulose and the lignin. But I always like to think like, oh, some of those spores that are out in space that were maybe on that meteor, like they came in and they were more highly evolved because, it's more extreme out there. But what are they eating? What
Yeah.
cellulose lignin are they eating? What
They're
kind of
amazing.
foods do they have?
You can get them. You can get some to eat styrofoam. I mean, it's just they tremendous capacity to
Yeah.
digest matter or things. Somebody asked me, what am I most excited about? I'm excited about everything, but I am excited about the work with NASA. We sent
Yes. Tell
we
us more
sent
about that.
we sent the black fungus out to space, and NASA is very interested in potentially using melanin for shielding. The big problem that we're going to have as a species is radiation.
Hmm.
And that's, you getting us to Mars, you can make a ship, but how to shield
Hmm.
humans in space for years?
Yeah.
It's hard.
Sort of like maybe they're trying to shield, like that's why they're
Exactly. So
growing, like they're doing their
there's a
job.
lot of sci-fi thinking, like for example, that you can
Hmm.
have a double hole and you can grow the mushrooms in the outer hole, black mushrooms, and that will give you a lot of protection and you don't have to send lead up to orbit. I mean, you
Mm-hmm.
can only send one spore, right? Then can you just grow the thing in
Yeah,
space rather than send tons of lead. And
but make like a nice shield of,
oh, right.
gosh, what would that look like materials wise?
Well, I think that there's a lot of interest in growing them on surfaces and things like
Hmm.
that. They can directionally grow.
Hmm.
So they're great discussions. It's a world
Yeah.
out there for the taking.
Yes, yes. And I love as I mentioned at the beginning, we're building out this kind of little micro lab. It's actually in a I kind of wish it was a 1969 air flight, like little canned ham, you know, the little caravans that people have. We're making a tiny little lab to start. It's like our just getting started. Kind of wish it was 68 or 69, the year that fungi became it's officially a kingdom. But we want to try the starburst test.
Yes, you heard about that. He's so clever.
Yes.
He basically
And
just
Texas
put it
is
in.
so hot, so we're gonna
Yeah,
find
and
some
you take it
weird
out
stuff.
and starburst you know, goes into solution, and then you take that and you work with it. But that's how you pull the fungus out of the
Yeah,
sidewalk. You're not gonna
maybe
go
make
with a
it a
Q-tip.
game.
You can't
We
go
can
with a Q-tip. I know you better put starburst on it.
I think we can make it a game, like who can grow the most melanated,
Out of
like
the
who can find
starfish.
the hottest spot in Texas with their starburst and then grow the most melanated bacteria, fungus. But yeah, that was real
Well,
fun.
great pleasure visiting with you guys.
Yes,
Thank
yes,
you
and
for
we'll
having me, and I answer emails, so feel free to
okay,
write.
great. Yeah, I appreciate you so much visiting with us tonight and sharing all the cool research from your book and Sherry's iPad. There's a What If Fungi Win coming to you.
Well, thank you, Angel, and good night to all of you, and keep it up.
All right, have a good one.
Take
Good
care.
night,
Bye-bye.
everyone.